Hey, ladies and gentlemen, welcome back. I had to come back inside because it was so hot outside that my computer started overheating. So here it is, but I'm keeping the sunglasses on because I feel cool. So let's go into what are we talking about today? Passive transport? So let's get ourselves into passive transport and we'll talk about how and why things come and go out of your cells. Before we get into actual passive transport, there are a couple keywords we need to understand in order to understand how all this stuff works. So the first one is a vocab word called concentration gradient. I know people get this confused all the time, probably gonna hear me saying it a lot. Concentration gradient is the difference in concentration of something throughout a solution. That is your keystone definition. What it basically means is there's more stuff on one side than the other. And I told you in vocab that there is a basic rule. And that basic rule is nature loves balance, right? If things are not balanced, if things are not spread evenly across whatever you're in, then they'll get spread evenly. And here's what I mean by that. Here we got a beaker of water and it has a bunch of what we'll call solute, that is things dissolved in water. We'll say this is sugar, right? So the sugar is spread evenly and nature is happy. But if things are not spread evenly, here we go. There's our lump of sugar. Each little yellow circle is going to be a particle of sugar, sugar molecules. So when we put that into water, the water starts breaking the molecules up and they're going to start moving around because of physics. There's no energy involved, all right? I'm not heating this up, I'm not stirring it. Just by nature, the sugar molecules are going to start moving. Right now, what we have is called a concentration gradient, where we have a lot of particles tightly concentrated in one spot, but not in the other spots. And since nature wants balance, nature wants things to be spread out evenly, that's what's going to happen. Right? The basic physics of this is that all the electrons and all of these atoms are bouncing off each other, right? And that's giving them push. So just by those natural laws of physics, these things are going to naturally spread out evenly from an area of high concentration to an area of low concentration up here. They'll spread out until everything is evenly distributed and nature is happy. That's diffusion. Diffusion was caused by this concentration gradient. Nature doesn't like a concentration gradient. Nature fixed it, and now it's happy. It's spread evenly. And this concentration gradient is what's going to make passive transport work. It also has to do with some active transport, which we'll get to in another video. Speaking of active transport in another video, this is a key word that's going to be important for that, vesicle. This is another vocab word. It is a bubble made out of lipids, meaning membrane, a bubble made out of the cell's membrane, and it's going to be used to forcefully transport stuff. That does require energy. So that is active transport. Vesicle is the bubble that's going to be used for that. We will get back to this in another video. I probably gave you some kind of handout that you're writing on right now. Do not write this because this is just going to show up again in a second anyway. There are three kinds of passive transport: diffusion, osmosis, and facilitated diffusion. Let's break this down really quick, all right? Remember concentration gradient, things being in a very high concentration and being more concentrated here, a little less concentrated, and then not concentrated at all, right? That is a gradient, a gradual change in concentration. Diffusion is how nature fixes that. It's the particles spreading out evenly. That's diffusion. I'm going to jump down to this facilitate diffusion. Facilitate just means to help. So it's diffusion with some help. We're going to have a protein. Maybe something is way too big to diffuse. There's like a barrier in the way and it can't get through the barrier. So there might be a doorway. That door is helping diffusion. That doorway is facilitating diffusion. We'll get back to this. And sometimes with osmosis, diffusion can't happen at all. Something is blocking the sugar from moving. So if sugar can't move one direction, osmosis is our backup plan. Water will move in the other direction. How does that help? We'll talk about it when we get to that. Let's go into detail. Diffusion. Movement of particles from an area of high concentration to an area of low concentration. Guys, we just talked about this. It's just this happening. Particles are high concentration. Diffusion is when they spread out. Concentration gradient. Nature doesn't like it, so they diffuse. It means to spread out. They go from high concentration to low concentration. Now, sometimes there's a cell membrane in the way. Remember, guys, this is all about getting in and out of your cells. All these particles are outside the cell, and they need to get inside the cell. In this case, we'll say that this is oxygen molecules, right? You are breathing a whole bunch of oxygen, and these things are trying to get into your cells because we learned in last chapter, oxygen needs to get into your cells to get to your mitochondria to give you energy, right? So we need that oxygen down here. So how's that going to happen? As they are diffusing, there's a high concentration, there's a low concentration, so they're going to spread out. Well, if it's a small enough particle, they're just going to move through the lipid bilayer. That's right. They'll just squeeze right on in, right in between those lipids. And life is good. And that's just going to keep happening over and over and over again. They're just going to go from one side of the membrane right through the lipids and into the other side of the membrane until everything is nice and evenly balanced. So here's our example. We have a bunch of particles inside the cell. Let's say that is carbon dioxide, CO2. We want to get rid of that, right? There's a high concentration inside, a low concentration outside. It means there's a lot inside and only a little bit outside. Nature doesn't like that. Nature wants balance. So they are going to diffuse out of the cell. Another example, oxygen. You just took a deep breath. Now there's a whole bunch of oxygen outside the cell, only a little bit inside the cell. That is not balanced. Nature doesn't like that. So these are going to move inside until everything's balanced. Nature loves balance. Diffusion is nature getting balance. Osmosis. Osmosis. The movement of water from an area high concentration to an area of low concentration. That sounds a lot like diffusion, doesn't it? Here's the difference. Diffusion is when it was particles moving, right? The thing dissolved in water. You dissolved salt in water, and it's the salt particles moving. The water's not moving, the salt is. That's diffusion. Osmosis, it's when the water moves. So movement of water from an area high concentration to an area of low concentration. If the salt can't move, the water will instead. It's a backup plan. Here's what I mean by this. This video is going to go through diffusion, osmosis, and dialysis, which I didn't talk about, but since it's here, I'll show it to you quickly. So diffusion is what we talked about. The blue spheres are water molecules floating around. They're already spread evenly. And the orange is some kind of solute. We'll say it's sugar dissolved in here. And there is a concentration gradient. There's a high concentration here and a low concentration here. Nature doesn't want that. So they are going to diffuse across the tank until they're even. That's diffusion. Sometimes those particles can't move. We put in what's called a semi-permeable membrane, just like your cell membrane. Semi-permeable. Permeable means things can come and go. Semi means some of them. Some things can come and go. In this case, water can very easily come and go, but the sugar is too big. Check that out, this zoom in here. Water molecules are small enough to get in, but the sugar is way too big. So what's going to happen here? These sugar molecules desperately want to spread out even. They want to be as even as the water is even, but they can't because the wall's in the way. But nature is going to make them spread out anyway. So what's going to happen is a backup plan. If the sugar can't go left, then the water is going to go right. And that is going to increase the water level on the right side, allowing the sugar molecules to spread out more. And that will keep going until the water starts spilling out the side. And even though we're not talking about today, just because it's here, dialysis is when it's not the water moving. Do you have more than one thing dissolved in there? And dialysis is when we're going to make some things spread out even, the little things, but not other things. It's like a filter. This is what your kidneys do. Your kidneys are going to allow some things out of your blood and into your urine, but it's going to keep your blood cells in your blood vessels, and all the waste gets to go through. But that's not what we're talking about today. You take a look at this. This came right out of your keystones. All right, this is an example the state gave. I want you guys to focus on this because I'm told that this question does come up on the keystone test. All right, what we have here is yellow food color and blue food color inside of a dialysis tube. This is the same semi-permeable membrane we just saw in that fish tank. Right? So yellow food color water, and then in this semi-permeable bag, blue food color water. And after 3 minutes, they both turn green. Lucky for you guys, I went ahead and colored that in for you. So we got yellow food color and blue food color, and after some time, they both turn green. What happened here? Well, what happened is, those of you who know anything about art know that if you mix yellow and blue, they turn green. So if there is green on the outside, that means this blue water went through and the blue mixed with the yellow and turned it all green. And if there's green on the inside, that means this yellow went in the bag and mixed with this blue, and that turned green. Now, why is that important? Because some things aren't going to go through. This is not from the keystones, but it could be. They could change the question around. So I changed the question around. Here we see yellow and blue, but this time it only turned green in the bag. So what happened here? Why is this still yellow? Because the blue did not leave. That means this, whatever this blue material is too big to get out of the bag, but the yellow did get in. The yellow mixed with the blue in here and turned green, so that means the yellow is small enough to get in. That's what the keystone question is going to be based on, size. The blue is too big to get out, but the yellow is small enough to get in. So since the yellow got in, it mixed and turned green. And then I reversed it. This time, the blue stayed blue and the yellow turned green, which means the yellow was too big to get in, so it stayed blue. But the blue was small enough to get out, and the blue and the yellow turned green. Okay. All this tells us is size of the particles, right? If it's too big, it stays put. If it's small enough, it moves, it diffuses. In this case, this was water, so osmosis. Here's another one. Yes, I'm spending a lot of time on this page because I'm told from past students that these come up on the keystones. So let's zoom in. Can you read this better? All right, what you got here is not 4 different bags. All right, this is all the same bag 4 different times. This is the bag by itself, and inside the bag, there is 95% water and 5% starch. Sugar, guys, glucose, okay? Carbohydrates. So 5% of the contents is starch, the rest, 95, is water. If you put this bag in a beaker, like this beaker here, and the water in this beaker is also 95% water, 5% starch, that means it's the same inside and outside. The contents of the bag are the same as the outside. They're already balanced. Nature is happy. Nature got its balance. Nothing is going to happen. The bag stays the same. But what if you put it in pure water? 100% water in the beaker. That means there's 5% starch in the bag. We have a concentration gradient. a lot of starch in the bag, not a lot of starch outside the bag. The starch wants to get out. The starch wants to spread out evenly. It can't get out of the bag. So instead, water goes in. That's osmosis. Water goes in, the bag gets bigger, and the starch can spread. Remember, guys, backup plan. If the starch can't move out, the water will go in. Other way around. If there is 80% water and 20% starch in the beaker, that means there's more starch outside than inside. More starch in the outside water than there is in the inside water. So there's a high concentration of starch and a low concentration of starch, which means concentration gradient. High, low, the starch wants to spread in. But again, if the starch can't get in, then osmosis will happen. The water will come out. And the bag got smaller because it lost water. That's what happened. There's a keystone question, and it could ask you to explain what is happening. It may give you a situation and ask you to pick which one represents the situation. So understand this. If you don't understand this, contact me. Facilitated diffusion. Guys, this is super easy. All right? Don't get bogged down by the bigger word. Facilitated just means with help. So it's diffusion. Plain old normal diffusion, but this time with help. Right? You see that there are particles outside the cell. A lot of them outside, only one on the inside. What we have is a concentration gradient. So nature wants these things to get in here. But look, they're too big to get through the membrane, so they're going to need a doorway. That's what facilitate means. Facilitate means with help. This doorway is made out of protein, and it is going to help the particle get in. By help, I mean it's just going to be an open door so the particle can get itself through until it's evenly balanced on both sides. This did not require any of your energy. Did you see any ATP in here? No, you did not use any of your own energy. So this is still passive transport, meaning nature just happened. You did no work. So the particles can't go through the membrane on their own, so proteins will facilitate, will help the movement along. Nice and easy. If you did use your own energy, then it would be active transport, and that's the next video. So we'll worry about that next time. Right now, we're going to call it a day. Go back in Google Classroom, see if I left you any work to do, and I will see you in the next video.